Bismillahirrahmanirrahim. So today we'll be moving on and we will be starting chapter 2 according to the IGCSE syllabus for Cambridge, and that is titled Organization of the Organism. This is chapter 1 in O-level, and that is titled The Cell, plain and simple. Okay, now the way this chapter goes is they talk about the cell and its structure and its features and functions, and then they talk about organization of cells into more complex structures, and then after all of that they come down and they talk about the microscope, right? The instrument that helps you see cells, right?
Yeah.
I'm going to do that first. I'm going to talk about the micro. It's not at all a complicated topic. They just talk about, okay, fine, this is right, an instrument that we use to observe cells. Now, before that, right, just an introduction is what is the cell, right? So it is the fundamental M unit of life, right? So we being, obviously larger organisms, we've just come out of talking about organisms. We've talked about unicellular organisms and multicellular organisms and the different kingdoms and everything, right? And one thing, like as I just mentioned, you've got organisms that are unicellular, which means they're just one cell, or they are multicellular, which means they're made up of many cells, right? And so, right, the cell is the fundamental unit of life, right? So we being so unicellulars are just like a single cell, right? And they're alive and they carry out their functions. All the functions of living organisms, again, that we've already covered, right? The Mrs. Gren thing. And right, multicellular organisms are made up of, as the word says,
Many different.
Yes. So we're multiple cells, right? Those multiple cells can be hundreds to thousands to millions or billions, right? So we have like billions of cells in our body, right? And so normally I would introduce us, okay, fine. So we are an organism, we function, we have like what we know as skin and bones and organs and different types of organs, and those organs perform different functions. And the question is like, what are we actually made of? Is like skin like a thing of its own, or is it made up of something more basic, right? Our bones, are the organs like, are they like, the heart is a thing that people have, or is it made up of something more basic? Can we study it on a more basic level? And when we do study it on a more basic level, we come across the fact that there are something called cells that we are made up of, right? And then when you talk about cells, right, there is no typical cell, right? So there are certain typical features of cells, but there's no typical cell, right? And so there are different types of cells, and those different types of cells come together and they form more complex structures and so on, and then we end up with the organism. So that's just an introduction, right? I'm not talking about any of the features of the cell or any of the functions of those features. I just like, okay, fine, this is the focus of the chapter. Now, how do we get to know this? Like, how do we observe the cell, right? How do we see the cell so that we're able to study it, like what's inside it? And for that, we have, right, as far as this, like the O-level syllabus is concerned, we have the light microscope. Right? So when like a piece of an organism, for example, a plant shoot, right, is observed under, right, this light microscope.
What's a plant shoot?
Plant shoot is basically the stem part that grows with the leaves.
Oh, okay.
Okay, that looks nothing like leaves, but you get the point.
Yeah.
Okay, so let me just screenshot this thing. So here you have a light microscope. Can I fit it in here somewhere? Can I make it smaller? Should be able to make it. Oh yes. Okay, so this is a light microscope. I'll just remove this tab so it's more clearly visible. And there's another screenshot I need. Going back. Just a minute. Obviously, I can't prepare these screenshots beforehand. And this guy. That smaller. And okay. So what we have over here is the light microscope, as you can see on the left. And if you're able to see it clearly, right, the base is a stand. Then over here you have a light source, so there's probably a bulb or something. This is the stage where something called a microscope slide is placed. So microscope slide is a rectangular piece of glass, and what you do on it, like in the center, you place what's called, so this is a slide, and in the center you place what's called the specimen. So something, some like, very small thin part of a living organism. Could be small and thin. And what you do on top of it is you place a cover called a cover slip. And then you place it over here on the stage. Okay, so what happens is light from the light source, so the light source is right below, and the light travels up through the slide, through the specimen, right? You place it such that, right, the specimen is over the hole over here, so that light can pass through it, and the light passes through that and then goes into the lens, right? And from there, over here, you should be able to, I don't know, observe it. That's supposed to be an eye.
Yeah.
Right? So that's how you, and when you observe it, so for example, when we take a plant shoot, as I mentioned, this is what it looks like. So, right, you can see like tiny compartments.
Yeah.
Right? That's what a piece of an organism looks like. Now, obviously, not necessarily exactly like that. It depends on what part of the organism you took the cell.
But similar, like similar to this.
Like, I wouldn't even say similar. Like, look at this image. Does this look similar?
Oh no, that's a plant shoot as well.
No, this is from a kidney.
Oh, that's a cells of kidney.
Yeah. Right, so I'm gonna place it over here. Right. So this is these are kidney cells.
Okay.
The point to note over here is the compartment. So you can see that they're like individual units. Can you make out that difference?
The circles are the different compartments. Yeah.
Not the circles. Let me sort of make this bigger.
The circles are the cells, right?
No. Wait. Now, within the circles, you
Oh,
notice
it's like
this
it's
part,
like yeah,
this like I mean, for lack of a better analogy, pizza slices,
yeah.
or maybe like the pieces of an orange when you peel it.
I remember the diagram you showed from the textbook. It was similar to this. I forgot.
Like, can you make out these individual?
Yeah, 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 yeah.
So those are the cells,
Oh, okay.
right?
Okay, okay.
These individual pieces, these individual units. These are the cells,
Okay.
right?
Okay. Okay.
Over here as well, right? I mean, they're
Yeah.
smaller. Obviously, they're more cramped, so they're not clearly visible. So those are
Yeah.
cells, right? So the difference being that, which part of the organism, like which organism and which part of the organism are you sort of studying under the microscope. That obviously makes a difference,
Okay.
right? But the point is, one thing that you see is, right, when you look at it under a microscope, you see that it is organized in these individual units, and those individual units are the cell. Okay.
Okay.
So that's how we got to know, oh, cells. I mean, we actually got to know about cells even earlier than that. There's a history about Robert Hooke and studying cork and stuff like that. But, I guess this much is enough.
Yeah.
Now, right, some details about the microscope that you need to know. Uh, right, some details are mentioned in the text as to its the different parts of the microscope. Right, I went over it only briefly. Right.
Okay. Yeah.
So you can go through in more detail in the text, and of course, if you have any questions about that, then ask. Okay. Now,
Yeah.
magnification. So obviously, when you observe, right, the observed image through a microscope, eyepiece is much larger than the actual size of cells, right?
Yeah.
So what you're seeing is not how big they actually are, they've been magnified, right? So the initial magnification is times 10, for example. The x represents times, and then it could be anywhere between, right, times uh, 4 and 10 and 40 or whatever, right? So for example, it's times 40. So when you put these together, these two together, and you're getting a total magnification of times 400, because it's first, like, it's obviously 1, it's magnified times 10, so it's times 10, and that times 10 is magnified times 40. So basically you're doing 10 times 40, which is giving you 400,
Yeah.
right? Or
Okay.
1 into 10 into 40, which is giving you 400. So what you're
Mm.
observing is 400 times larger.
Oh, okay. So more zoomed in than normal.
Yes, it's very much zoomed in, and this is like the magnification of a school grade light microscope, right? Advanced light microscopes, uh,
Go much deeper than that?
they go up to 1500,
Oh shit. Okay.
right? So that's like around 4 times that,
Okay.
right? 4 times 4 is 16. Uh, so they go up to like 1500 times magnification. So that is considerable. Now, the interesting thing is that despite going up till 1500, right, the finer features cannot be observed. still, right? So there's something called the electron microscope that allows scientists to see the finer features of cells. It's not in your syllabus, it's not in your O level or IGCSE syllabus. That's something that comes up in A level. So if you decide to do A level biology, we can talk about that then. But okay, this. Now, what you need to know about microscopes, other than its different parts and how they work, is how to calculate magnification. One thing. So magnification is, right, the observed image size divided by the actual size of the specimen. So magnification equals to observed image size divided by actual size of specimen. Let me just
Okay.
make that line go all the way through. So for example, you have, right, you know that the, now for this, in fact, before I give you an example, and you can manipulate this, and you can go like, oh, actual size equals to observed size divided by magnification. So that's just manipulating the equation. Now, to be able to do this, you need to understand what scale is the observed and the actual size, right? And for that, I will show you this, right? It's all measured in meters, but how big this, and this should be easy because, right, you've done uh, you've done physics, right? The conversions, right? So here, now to clarify, right? This one that's given over here, this is 1 micrometer.
Okay.
Right? The standard they're using is 1 micrometer. And so something that's a bit larger than 1 micrometer is a bacterium. And how much is 1 micrometer? Right? 1 micrometer is 1 into 10 to the power minus 3. Oh, sorry, that's 1 into 10 to the power minus 6 meters. Right?
Yeah.
So a micrometer is 1 into 10 to the power minus 6 meters to begin with. And the reason I'm converting that to meters is because meters is the SI unit of length. Wait, I think this is being hidden behind my, yeah. So let me just, whoa, okay, no. Let me just put it here, over here, and sorry about this. Okay, so this is 1 micrometer, right? And 1 micrometer, so that's how it's written, is 1 into 10 to the power minus 6 meter. And the reason we're giving it in meters is because that is the SI unit to measure any length. Okay, now what you need to know, right, is when you are doing the calculations, you will be either measuring in centimeters, which is 1 into 10 to the power minus 2 meter, or you'll be measuring in millimeter, right, which is 1 into 10 to the power minus 3 meter. What you need to know is converting one to the other, right? So 1 centimeter is 1 into 10 to the power minus 2 meters, 1 millimeter is 1 into 10 to the minus 3 meters, 1 micrometer is already given over here. And so what you need is, when you're working with this, right, this should be the same, right? They should be in the same like power, or the same standard form. Is this making sense?
Yeah.
Right? So for example, you have, right, you observed an image of a cell, and they asked you to measure its length, and you go like, okay, fine, I'll measure from here and change the color. I'm going to measure from here till here. Right? You take that measurement and you go like, right, this is, for example, 37. Now, what is it? 37, either you go millimeters or you go 3.7 centimeter. Right? Now, which one? And for example, right, they said that the actual size is either they've given in millimeters, or for example, they've given it in they've given that in micrometer. So they say the actual size is, for example, 1 into 10 to the power, well, not 1, it's, for example, 6 point, I don't know, 6 into 10 to the power minus 6 meters, which is basically 6.6 micrometers. Are you following?
Okay.
Are you following?
Yeah.
Right? So they've given like, okay, fine, this is 6.6 micrometers. So what you need to do is you need to convert, right, 6.6 micrometers. You need to convert that to standard form. Then you take the 37 millimeter, you go like, okay, fine, 37 millimeters is 3.7 into 10 to the power minus 3 meters. Now you've got them both in standard form.
Yeah. Okay.
Okay. Then you can apply the formula. Okay, fine. Observed image over actual size. So observed is 3.7. So magnification.
So it will be 3.7 over 37.
No, 3.7 into 10 to the minus 3 meters. You've got them in the same unit, right?
Yeah.
Over 6.6 into 10 to the power minus 6.
Okay.
Okay, now what I'm going to do is I'm going to just for the sake of making this calculation easier, I'm going to make this 33, just so I can like, right? So this is 33 or 3.3 or 3.3. Oh yeah, over here. So I'm just like doing that so I can do it very quickly. 3.3. And so what you have, right? This is 2, right?
Yeah.
And then you have, right, you do the standard form thing, and what you're going to end up with is 1 over 10 to the power minus 3, and that can be converted, right? So what you're going to get is 1 over 2 into 10 to the power minus 3, right?
Okay.
If I'm not
Yeah.
mistaken, this converts to 2 into 10 to the power 3. Does it not?
I gotta check for that. Wait.
which comes up to 2000. So your magnification is times 2000.
You know, 1/2 multiplied by 10 with the minus 3 is 0.0005.
Wait, let me check as well. 1 divided by 2 into 10 to the power minus 3.
Yeah.
Oh, sorry, it's 500. So I was trying to do this mentally. I did it very, very wrong. So it's times 500, not times 2000. Right?
Yeah.
And so that is your magnification.
Okay.
Right? It doesn't become 2 into 10 to the minus 3. It becomes, yeah, this is the mistake I made. Right? Is it becomes 1/2 into 10 to the power 3. Right? So just the power is going to be moved to the numerator to make it positive. And so that's basically half times 10 to the power 3. So that becomes times. So that is your magnification. Right? So whatever you were observing, whatever cell you were observing, was 500 times larger than what actually was. Now, I mean, were you able to follow this? Did this make sense?
Yeah.
Are you sure?
Yes.
Okay. So obviously, when we like the magnification questions in the paper are not super tricky, right? The only thing you need to keep in mind is that, right, you need to keep them in the same standard form, like unit, right? So same unit or standard form, right? So you converted both of them in meters. You did the calculation. You could have kept both of them in millimeters. You could have converted this into millimeters. You could have converted into centimeters. As long as they're both the same, you're going to get the same magnification,
Okay.
right? So whatever you're measuring, the actual size and the image size, right, should be right in the same unit, right? Millimeter, centimeter, micrometer, meter, whatever you're doing. As long as you have those the same, you're going to end up with the correct answer. Now, another way this can be given is you're asked to find the actual size, right? And so actual size is image size divided by magnification, right? And so, for example, you have an image size of 6.9 centimeters, right? And they tell you that it has a magnification of times 350. And so what you're going to do is you're going to do 6.9 divided by 350, right? And
Yeah.
what do you get? And this is the only, I believe, part in the entire biology syllabus where you need a calculator.
Oh, okay.
Formulas. So divide
Yeah.
by 350, right? And that gives you
0.
0.019714.
Yeah.
Zero. Now this is not our final answer. 714 centimeters. Then they ask you, oh, give the actual size in centimeters.
Isn't that in centimeters?
Oh, sorry, micrometer.
Okay.
Right. So when this is in centimeters, this is 0.019714 into 10 to the power minus 2. right? Meters.
Yeah.
And so we know that micrometers is 10 to the power minus 6. So what we need to do is we need to shift the decimal place. How many points to the right?
2.
4.
Oh, okay. Yeah. Okay.
1, 2, 3, 4.
So it will be
Right, so that would make it, yeah,
197.14.
so that would make it 197.14, right, into 10 to the power minus 6 meters, or right, 197, sorry, 197.14 micrometers.
Okay.
Make sense?
Yeah.
Acha. Now, another thing to keep in mind is, right, significant figures. You're familiar with significant figures. Yes?
Okay. Yeah.
Right? So you probably won't be giving an answer like that. You'll be giving an answer of 197 micrometers. You skip,
Yeah.
right? Have you followed along?
Yes.
Are you sure?
Yeah.
Right? So recap, right? Uh, Microscope used to observe, right, the structure of organisms, like the microstructures, so cells and stuff, right? And magnification, there's a formula: observed image size over actual image size. You can manipulate that to find whatever the unknown is. And when you are finding the unknown, you need to make sure that the sizes are either in standard form with, the SI unit of meter, or they are in the same unit, right? So we had something that was either in millimeter or centimeter. We converted that to standard form. We had this for micrometer, so 10 to the minus 6 and 10 to the power minus 3. We did our calculations, right? And we got a 500 times. Now, magnification is written as x and then whatever number, right? This basically means 500 times, right? So x
Yeah.
350 means 350 times. So there's no unit for magnification. Keep that in mind. It's just a number. We put x to denote like a multiple. So this, whatever the actual size was multiplied and shown bigger 350 times than it actually was. Okay,
Yeah. Okay.
right? And so we calculated that and we did our work, and like, oh no, we wanted micrometers. Okay, fine, micrometers. We're familiar with micrometers. It goes up to 10 to the minus 6, and so we converted that and got our answer.
Okay.
All right. And
Yeah.
obviously you'll have a calculator to do that, so it'll be easier.
Yeah.
Okay. Now, do we have time? So that was about the microscope, right?
10 minutes. Yeah.
Yeah. So they mention all this right at the end of the chapter. So okay, fine. We're talking about the instrument that helps, you observe cells. So let's talk about that first. Now, uh, there's a lot of very basic explanation in the text, and I'm not going to go over that. Right, you can just read it, and still, if there's anything that is unclear, you can ask. So how do we take samples of cells? Right, there are two ways of taking it. So for example, if you've got this piece, right, this is a piece of tissue or something, like not like the facial tissues, right, like a tissue of an organism, like a piece of an organism. And so there are two ways you can take the sample. Either you can cut across, like this. Okay, and this
Yeah.
is called a transverse section. Okay,
Okay.
or you can cut. So this is across, cutting across. So it's cut across, and this is called a transverse section. Or you can cut along its length. Okay,
Yeah.
this, right? So cut along length, and that is called a longitudinal section. Why does this matter? It matters because cells are actually three-dimensional. They're not two-dimensional like we draw on paper, obviously, right? So if
Yeah.
this is a cell, right? In fact, let's go back to the images that we used earlier, right? So this is a cell, right? Now, obviously, this is obviously a specimen either taken in longitudinal or transverse. It seems to be transverse. So what you notice is the way the specimen looks under the microscope will differ, right?
Okay.
For example, like you've got a cucumber, right? A cucumber is like obviously a long piece. So, right, you've seen cucumbers. I don't know, like this.
Yeah.
Now, if you're cutting it transverse, you're going to get the round slices.
Yeah.
But if you're cutting it like this, you're going to get longer slices,
Yeah.
right? And if you've ever observed, they look different, right? The round slices of cucumber, right? They have like the seeds in a circular pattern,
Yeah.
whereas the right the longitudinal section of cucumbers, they don't have the seeds in a longitudinal. The seeds appear to be like in a straight line,
Yeah.
even though they're taken from the same cucumber. So the way the sample, like the cells within the sample, appear in the microscope depends on the way the sample was taken.
So like whether you take it horizontally or vertically, the cells will be in different positions, right?
Exactly, because the cell is three-dimensional, and right, so you've got different stuff. For example, right, you have right the nucleus over here. So we haven't talked about these guys, right? And you've got some stuff over here and some stuff over here and stuff over here. So if you're going to cut across, right, one way or the other, right? Let's say that this is like a tube of cells. So when you cut across in the transverse, you're going to get like a whole cell. You're going to see the image of a whole cell, right? But if you cut across what you're saying horizontally, you're only going to get this part. You're not going to be able
Yeah.
to see this part. For example,
For example.
yeah, I'm just saying, like, for example, you will only be able to see this part of the cell and not the rest. Whereas if you cut, now, this doesn't mean that the transverse section is better than the longitudinal section. No, right? Both of them have their benefits. Both of them are taken for reasons, right?
Yeah.
Yeah. So it's again, what you're observing under the microscope depends on how the sample was prepared, right? What part was taken, how it was cut, how it was prepared, that will affect what you're able to see.
Okay.
That's the
Okay.
point. That's the only point I'm trying to make.
Yeah. Okay.
Okay.
Yep.
I guess we'll stop here.
Oh, okay.
Is all of this clear?
Yeah.
All right. So now that we know about the microscope, and we've mentioned a few things also, like the nucleus and stuff, right? So next class when we sit down, uh, right, we will, hopefully cover more ground.
Yep. Okay.
All
Sure.
right.
Yep.
And again, obviously, read the text, and if you got any questions regarding what we've done today, do ask. You'll find this information, the microscope, at the end of the chapter, and the part where I talk about the longitudinal and transverse section at the beginning of the chapter. So read the very beginning, like the first
Okay.
two pages or something, 2 3
Okay.
pages, and then read the end where they talk about the
So the
microscope.
beginning and the end of the chapter. Okay.
Basically, yeah.
Okay. Got it.
Okay.
Yep.
All right.
All right. Okay then. Allah Hafiz.
Love.